We basically, yes, in this slide, sorry, we were further focused that what we did, we worked on the 174 homozygous damaging SNPs, which I mentioned before. 104 homozygous damaging mutation. 174 mutations which were homozygous, present on both chromosomes, and those mutations were damaging non-synonymous mutation. And these we found that 14 were found found related associated with different diseases or phenotypic information. Some of these related to Pakistani population as well, before all that is previously known to be involved, to be reported from Pakistani individual. And out of that, this, for example, this gene, this is fucotin gene, which is involved in cardiomyopathy. That is, in cardiomyopathy is a cardiovascular condition, cardiovascular disease, and this defect in this gene is involved in cardiomyopathy, in cardiovascular condition. And there is another gene called as dystrophin, very famous gene, also involved in cardiomyopathy. So the mutation in this gene in this particular individual can be is also previously known to be involved in cardiomyopathy or cardiovascular individual. Beside some other condition which I'm not going into the detail. Say, for example, this gene, which is basically a zinc transporter, is reported to be involved in diabetes mellitus. So starting from the sequence and sequence analysis, we came out up to the disease. That is we came starting from the genotype, we discovered the phenotype at disease level, that these genotype can be involved in this particular phenotype, which is in this condition are different diseases. That is cardiomyopathy and diabetes and some other diseases. Although, of course, it needs more information, but I wanted to tell over here to you that this that is the analysis of genome sequencing analysis can provide us the information about the disease condition, or and the sequences or genome mutation can be correlated with different genomes, with different disease conditions. Okay. Next. Okay, now next we basically compared the genome sequence with Pakistani individual genome sequence with Indian genome sequence, revealed more than 100,000 SNPs shared between these two individuals, originated, or you can say that is which are both of these two individuals are from the same region. Okay. 24% of these shared, we call these mutations as shared mutations, were confined to coding regions. So these 100 and more than 100,000 sequences, more than 100,000 sequences, sorry, 100,000 shared SNPs are distributed all over the genome, okay, in the gene region as well as intergenic region. Out of these 100,000 SNPs, 24% are, that is 24,524 SNPs are confined to coding regions, different coding regions. 170 of these were non-synonymous SNPs and were located in 119 genes. 119 genes. Out of these 24,000 shared SNPs, 170 were non-synonymous or NS SNPs, and these 170 mutations were located in 119 genes. So what we did, we basically carried out a gene ontology analysis. Gene ontology analysis is basically gene classification analysis, you can say, that in which class these genes are classified, in which functional class these genes were classified. On the basis of gene ontology analysis, we found out that these 119 genes which contain shared SNPs are belonging to olfactory and taste receptor genes. They are involved in olfactory, that is, they are involved in smelling, and taste perception of bitter compounds. So this is very interesting to the people living in Indian subcontinent, because we in Indian subcontinent or South Asia, people are very much used to eat bitter compounds in spices and in other food material. So out of these shared SNPs, the genes involved were classified into olfactory or taste receptor genes. Okay, and this this was the first category. Another category was excuse me. Another category was the genes involved in carbohydrate metabolism and insulin secretion. Again, we know that the people in the Indian subcontinent are very much prone to have that is diabetic condition. So these genes are mutation in these genes are can be related to diabetic condition or carbohydrate metabolism. Another type of class, another class which were involved, genes involved in this class were involved in immune response. So as that is by analyzing two individual genome sequences from South Asia, from India and Pakistan, we basically able to found out some genes which were specifically involved in different functions related to metabolism and other biochemical pathways. Okay. Now, at the end, the conclusion. So, in the conclusion, what I can mention over here, that we sequenced a Pakistani individual genome at 25x coverage, which was significantly more than the sequencing carried out by 1000 Genome Project. The refined Pakistani individual genome data showed an array of Pakistan in specific SNPs not present in other databases containing SNPs and indel sequences. Okay, novel coding variants described in this study can be correlated with the health status and phenotypes over the entire human span. Okay, and uh, several of the disease associated SNP has already been reported in Pakistani individual, if not Pakistani population, we can say Pakistani individual. So these are some of the conclusion of the genome sequence and variant analysis of an individual. Okay. Next is excuse me, yes. Gene ontology analysis or gene classification analysis of genes containing PARC-ND and shared SNPs revealed interesting correlation. The present data can be helped. This study was provide further information for further genetic variation studies of ethnically diverse Pakistani population. As we know that Pakistani population is not a homogeneous population. It is ethnically diverse population from different regions, from different provinces of Pakistan. And so this study is a part of the so-called Human Genome Variation Initiative of Pakistan, in which we want to carry out genome sequence analysis of other individual from other part of the country. Resulting genome sequence information represents an important contribution to our knowledge of the genetic diversity of South Asia. Of course, said that this type of studies can provide us valuable information regarding the genetic diversity of South Asian subcontinent. Okay, so as the result of this data was published in Journal of Human Genetics. I'm sorry, the slide over here is blurred. So this is the journal of this paper was published in 2013 in Journal of Human Genetics in 2013. I will that is upload the PDF file of this journal into the Google Classroom. You can read complete paper from there. And this was basically as you see the title of this paper was the complete genome sequencing and variant analysis of a Pakistani individual. Okay. And I was the first author with Yang from China, and we collaborated with Chinese genomics center named as Beijing Genomics Institute, BGI, and my student Asifullah Khan and Huma Asif and Sana Sharif were part of this project, and this paper or this project was supervised along with me by Yong Zhang, a very reputed scientist in China, and Chinese scientists, and my group members when I was in the International Center for Chemical and Biological Sciences. My students were part of this work. And yes, next is so this is, I think,